is at the core of kidney function. When we talk about kidney failure, we're mostly talking about the failure of this filtration process. Right? That's usually what is going to be affected first, either through back pressure, as I mentioned, or high blood pressure, something like that, or clogging from like a massive amount of protein in your blood or something like that. That can cause acute failure. Right? What we're talking about then is the rate at which filtrate is produced, the glomerular filtration rate. And there are a few things that affect this. First is the permeability of the filtration membrane. How easy it is for material to move across that permeability. We know that there are holes, certain size, right? That the fenestrations prevent the cells and large proteins from passing through, and then slightly larger proteins can't get through the basal lamina, and then finally the filtration slips between the pedicels of the podocytes. They only allow very small molecules or very small polypeptides to go through. Okay? But this is somewhat flexible, somewhat variable. If the blood flow increases to the glomeruli, they can expand, kind of stretch, and that can make the membrane a bit more permeable. Right? And this is useful to get rid of extra volume if your blood volume is too high. This is also regulated in some ways by the position I'm so sorry, move on to the next one. The next one is filtration membrane area. That's the size of that filtration membrane. Okay, now you think that would be fixed, right? You've got your glomerulus, I always kind of represent it as a fist. You've got your glomerulus, it's got a certain length, certain surface area around each one, but it is somewhat variable. Because remember those cells in between, the mesangial cells? Those have a contractile property. They're not technically muscle cells, but they do have a contractile property, and they can constrict, so that glomerulus can get a little tighter. Now imagine that my fist is the glomerulus. If I make a very loose claw-like fist, then filtrate can flow out in between my fingers. But if I can squeeze it down, now it can't. So the membrane surface area is effectively smaller. Okay? And we'll talk about the role those mesangial cells play a little bit later. And then lastly, we have net filtration pressure, and obviously this one is the easiest to change because you have control over the rate at which blood is flowing into the glomerulus. You have two arterioles. You can also have control over the rate at which filtrate is flowing theoretically, but you don't really regulate that, right? You regulate the rate at which it's being produced by regulating the pressures pushing it out. Now there's two ways that we do this. One is familiar, it's the myogenic mechanism. This is something we talked about in the context of smooth muscle before. And if you stretch smooth muscle, it tends to pull back, okay? And this happens with the blood flow to the glomerulus as well. Let's say you have vasoconstriction somewhere, and so more blood is directed into the kidneys, just because it's going to go anywhere that you don't have constriction. That will stretch this afferent arteriole, and it will reflexively constrict back and reduce the blood flow. Okay? So that's the myogenic mechanism. Very simple, or at least something we've discussed before. The other one is a bit more complicated, and this is tuboglomerular feedback. And this takes advantage of the fact that the distal tube, one of our pictures that we use says this is the distal convoluted tubule. Another one says it's the ascending limb of the loop of Henle. I'm sure there are people that get fist fights over that at scientific meetings. Probably really if it's just glomerular, it's probably technically the loop of Henle. But we don't care. It's the other end of the tube, okay, or close to the other end. This passes back. So this is where the filtrate enters the tube, and this is close to where it's at the end, close to the end. So what's happened in between there is that reabsorption and secretion, or most of it anyway. And so the contents of the filtrate here are different than there. This has everything in it, this has much less. You have a set of cells that line this tube, and they're called the macula densa. We've seen the word macula before. It just means place or spot. Dense, of course, means dense. And we see here the cells are especially densely packed. Okay? And what these cells are doing is monitoring the contents of that filtrate that's passing by them. They are monitoring it to determine if the process of reabsorption / secretion, but really it's reabsorption, is going well. Now, most everything, and we'll look at a chart of the reabsorbed substances in a moment, but most everything is being reabsorbed here. So what do you look at? What do you measure to determine how well reabsorption is going? Well, of course, sodium, being the dominant extracellular cation, is a good target, because we reabsorb a lot of sodium, right? But we don't reabsorb all of it. So there's still sodium in there. So we can measure the amount of sodium, and chloride actually, because chloride and sodium go together with by electrical attraction. So we can measure the amount of sodium there. And if that amount of sodium is too high, one way that that could happen is if the filtrate is flowing too quickly. We're making too much. If we're making too much, it's pushing through that tube fast, and there's not enough time to reabsorb sodium, or as much sodium. And so by the time it gets to the end here, there's too much sodium in it. Right? It's like Lucy and Ethel didn't have time to pack the chocolates. And so what we can do in response to that is constrict these smooth muscle cells around the afferent arteriole. Now these are modified smooth muscle cells. They're called juxtaglomerular cells. And there is communication between the macula densa and them. It causes them to constrict. And that reduces the blood flow into the glomerulus. That lowers the hydrostatic pressure in the glomerulus. Reduces then the net filtration pressure and the filtration rate. Okay. On the other hand, if there's too little sodium here, it means that we've reabsorbed more than we normally would. And one way that that could happen is if the filtrate is being produced too slowly. And so the cells lining the tube have more time to work on it, and they reabsorb more sodium, and so now there's less here. Okay, so what would it do? It would cause these cells to dilate and increase blood flow into the kidney, or into the glomerulus. And that would increase the hydrostatic pressure in the glomerulus, which would increase the net filtration pressure, and increase the glomerular filtration. Yes?
How do you counteract that?
Huh?
Let's say someone just has too much sodium in their body. That plays a role in that.
Oh, there's another mechanism that would deal with that. Right?
Oh.
So, and we'll look at that as we get into this. But yeah, this is just the basic moment-to-moment mechanism. Yes, Sam?
So what is being constricted there?
It's the afferent arteriole, mainly, in this case. As we'll see in a moment, there's instances where you constrict both. Just reduce overall blood flow to the kidney, period, right? But in this case, it's the afferent. The afferent one has to control. If you were to constrict the efferent, the pressure in here would actually go up, right? When you put your thumb over the end of the garden hose, the pressure inside the garden hose increases. What we're talking about is downstream of that, okay? So when you constrict here, pressure in here drops. All right, now, if you have a big reduction in filtration rate, and it's way too low, then increasing it may be counterproductive for the whole body, right? Because let's say you've had a hemorrhage, you've lost a lot of blood volume. Blood flow to all your tissues is going down, including your kidneys, and this will drop the GFR. So tuboglomerular feedback would detect that there's too little sodium here, we're not filtering fast enough, and so now it dilates. Well, now you're pushing more blood into the kidneys. Maybe the brain or the heart could use that blood. The kidneys are a vital organ, but you can starve them just slightly to save the brain and the heart. So what do the kidneys do about that? Well, they have an override system, one that we've already discussed. If the blood pressure is too low, meaning the blood flow to the kidneys is too low, the filtration rate will be way too low. Now, I'm not saying this is only produced in extreme, but if it's overall too low, what the juxtaglmedullary cells can do then is release renin. It comes from them. And what does renin do? Converts angiotensinogen to angiotensin 1, which is then converted to angiotensin 2 by ACE in the lungs. And angiotensin 2, among other things, causes these to constrict. Overrides that. Okay? So it will reduce blood flow to the kidneys, reduce filtration, and that will help maintain blood volume to help maintain pressure. Okay, so here's tubal glomerular feedback. Something disrupts by increasing GFR. They detect increased delivery, right? So if it's going too fast, too much, there's increased delivery of sodium chloride. Afferent arteriole constricts, increased blood flow, runs back to normal. All right, so now let's look at other ways. Now I already just gave one of these away, but let's look at these. We also can regulate this using neural innervation, right? Because you're going to have sympathetic innervation to those smooth muscle beds around our blood vessels. And of course, as we've already discussed, there are hormones that directly affect this. So let's look now at the sympathetic innervation. And it kind of works in a two-phase fashion. Moderate sympathetic activity constricts both, and that results in a slight decrease in GFR. I'm not talking about clammo down to nothing, I'm talking about a slight decrease in both of them. So that gives you an overall lower flow to the kidneys, slight decrease in their function. More profound sympathetic activity, you get a larger constriction of the afferent, which would give you a larger drop in the GFR. So again, sympathetic will affect both of them, depending on how intense the stimulation is. All right, and then the hormones, as we just discussed, angiotensin 2, which results ultimately from the release of renin by the juxtaglomerular cells, causes constriction of the arterioles of the kidney. It's pretty selective for that. We know vasopressin, or ADH, is overall vasoconstrictor, but angiotensin 2 mainly affects these in the kidneys and some others, but these are one of its targets. Okay, so angiotensin 2 constricts that and drops the GFR by reducing blood flow. Now ANP, atrial natriuretic peptide, remember, is our one hormone we have to lower blood pressure. And mostly it works by blocking the effect of the others at some level. But here we can see one of its direct effects. What it does is cause these mesangial cells to relax. And that makes the fist go from being tight to loose. That increases the surface area of the filter, and therefore raises the GFR, and that would lower blood volume, increasing filtration, increasing the production of filtrate in urine. So let's go ahead and finish GFR, and then we'll take a break. Now, GFR is an important thing we have to measure because it is a good assessment of kidney function. one way to do this is to introduce something into the plasma and measure how quickly it is cleared from the plasma and ends up in the urine. And the rate at which it is cleared from the plasma will be directly related to the GFR, the glomerular filtration rate. It is important, though, to pick something that's going to go through the filter and not have any other way in or out of the body. So you would introduce this into the bloodstream, predicted amount of it. It would pass through the filter. First, it would be inert, not actually cause any harm, and it would be pass through the filter, not be reabsorbed, and not be secreted, so that any of it that you found in the urine had to have come through the filter. Okay? Now, this is still done in more advanced renal tests, but it used to be sort of the standard way to do it. You would give someone inulin, which I believe it is a polysaccharide. I can't remember the chemical nature of this molecule, but it is inert. Okay, it doesn't do anything. It's not insulin, it's inulin. It is inert, doesn't do anything. You introduce it, it passes through the filter, it ends up in the urine. So you give an injection of this or give it an IV, and then you collect urine over time and measure the inulin, and it's also easy to measure. Bonus, okay? And then by plugging this into some formula, you can measure the GFR. Okay? Very useful for that. This is still done if you need a really accurate, consistent way to do this. But it turns out there is a newer way that is much less invasive and easier. It turns out your body produces its own molecule like this. It naturally produces a molecule that passes through the filter and is not secreted or reabsorbed. This is called creatinine. And I'll show you the spelling of that in a minute. It is a metabolite of creatine. Remember, creatine is used to store phosphates, high energy phosphates in muscles. Okay, creatinine is produced in the muscles. There's a certain normal amount of it in the bloodstream, and it's going to pass through the filter and end up in the urine. So you can measure the amount of creatinine over time in the urine. But more useful is simply to measure the amount in the blood. Because if creatinine is elevated in the blood, it is one indicator that perhaps the kidneys aren't clearing the creatinine and probably other things as well. So elevated creatinine in the blood is an indicator of perhaps diminished kidney function. And okay, we need to follow up on that. Let's do a more advanced test. Okay, so this is like the holy grail of diagnostic tools is a simple blood test, right? Or perhaps even something less invasive, right? But blood tests are the big thing. Remember that Theranos controversy? That lady, what was her name, Elizabeth something? She fooled the whole world thinking she had developed all these advanced blood tests and only needed a tiny drop of blood and got billions of dollars in capital funds. Turns out it was all a scam. She's going to prison now. Because people want to have simple blood tests to tell them if they have something. Well, here we have one, right? We just measure creatinine levels in the blood. If they're elevated, then we can follow up. Because there might be some other reason for them to be elevated, right? But we could follow up and do a more precise measure of GFR. All right, so let's take our break, come back, and about transport challenges. And so we say basolateral membrane, okay? So that is, of course, going to be the most likely. Some things can move across between cells, and particularly if the transcellular absorption creates a strong diffusion gradient, things can slip through the gap-tight junction and be reabsorbed transcellularly, as we see here. And this is just to remind you, this is more relevant for the people I teach AMP2 that have not just had AMP1, but just to remind you of the difference. Active transport, we're moving against a concentration gradient, requires some energy source. Primary active transport is using ATP directly, and the transporter itself uses ATP. Secondary active transport is using an existing concentration gradient as an energy source, both of which involve some sort of molecule, some sort of molecular mechanism that has to move. And because of that, there's only so many of them, and they can only move so fast, and so we encounter this phenomenon of a transport maximum, right? We can only transport at a certain rate. Then we contrast that with passive. In the case of just regular diffusion, things either move through a channel or they move directly across the membrane because they can permeate the membrane easily. Or if they're too large to do that or too impermeable to do that, they can diffuse through a facilitated diffusion and transport. But again, this is passive because no additional energy is needed. They just move from high to low directly. All right, so with that, let's look at some of these reabsorbed substances. Okay, now sodium is going to be reabsorbed throughout the tube, as we'll see. Let's look and see how we're going to use that sodium reabsorption to power the reabsorption of things like glucose in the proximal convoluted tubule. We're going to have symporters of sodium and something else. So this is, we're using glucose as an example here. So we have the sodium glucose symporter, and sodium is slowing down its concentration gradient, and we're using that energy to move glucose up a gradient, okay? Now typically, glucose might be lower inside the cells because the cells are using it, and so we may not need active transport to move glucose into a cell because it would be going down its gradient. But remember what's happening here. We're trying to pull every last scrap of glucose out of this filtrate. And so it's building up inside these cells. So they're going to have a higher concentration. We have a low concentration out here, especially as we get towards the end and we have very little glucose left. So we need that active transport to go up the gradient. And then now once the glucose is in here, we can simply allow the glucose to move across the basolateral membrane via facilitated diffusion. Of course, all this relies on the sodium gradient, and so we have to use an ATP, primary active transport, to pump the sodium out. We pump the sodium out on the same side as we pump the glucose. That way we reabsorb sodium and glucose simultaneously. And we're going to use this same mechanism to reabsorb other things besides glucose. Amino acids, vitamins, phosphates, sulfates, other things like that. Okay. Now, this is something I've talked about before, but just to remind you, the reason people with untreated diabetes have glucose in their urine is because of that transport maximum. If you have too much glucose in the plasma, you will have too much glucose in the filtrate. And the cells of the proximal convoluted tubule will do their level best to transport that glucose. But there's simply too much, and the filtrate is only spending a certain amount of time in that proximal tube before it flows on down the tube. And so we have reached that transport maximum. And so we can't move it all out, and some of it ends up in the urine. And so we can measure that easily. As I've said before, glucose in the urine is actually not terribly dangerous. I guess it would increase the risk of a UTI or something. Well, it's what it indicates that's the bad part, right? That your cells are not absorbing the glucose. And that's causing long-term damage by glycosylating proteins, cause damage to blood vessels and nerves. But in the immediate life-threatening issue is that these cells are going to start using fats and amino acids to produce energy, and they're going to make keto acids as a side product, and you'll get acidosis. Right? Other electrolytes are going to be moving in. Some of them will move in transcellularly by diffusion. Okay? We're going to reabsorb quite a bit of urea as well in that way. But then I want to mention water. In the proximal tube, we are reabsorbing most of our water. And this we call obligatory water transport. Why obligatory? Because these cells always have aquaporins in their membranes, water channels. They're constantly present. And so as we move all that solute, all that sodium, all that glucose, vitamins, sulfates, amino acids, all these other ions, urea, as we move all that solute, the water follows by osmosis. Okay? So again, we don't have any active mechanism for pumping water molecularly. We have to move solute to move the water. Water follows solute. So this is known as obligatory water transport in the proximal tube because it always happens. Okay. Now secretion, as I mentioned, is also a mechanism that occurs here. Much less prominent though, as we indicated when I showed you the numbers, but nevertheless it is important. And so what sorts of substances are secreted? Well, one of them is potassium. As we noted, we do secrete some potassium. This occurs in the collecting duct. The principal cells, note principal cells of the collecting duct. And so what we have here is a sodium-potassium pump. And it's pumping sodium in, right? Sodium is being absorbed easily, right? Just flowing in through channels or through an antiporter, right here, and then just being pumped out, reabsorbing that sodium with potassium, okay? Now, a good bit of that potassium is just flowing right back out, back into the blood via the basolateral membrane, via channels there. But a fraction of it is flowing out and into the filtrate, and that's the secreted potassium. Right. Remember, we have a way to regulate water reabsorption with ADH, and we'll go over this again a number of times. But remember, ADH allows us to reabsorb water alone to lower our blood's osmolarity. Aldosterone allows us to reabsorb solute and water. Which solute? Sodium mainly, and the chloride comes with it, and the water comes with it by osmosis. So this allows us to bring in volume entire, so solute and water. So we're not changing osmolarity so much, but we are bringing in volume. ADH, you're bringing in water alone, that's raising volume, but it's also diluting the osmolarity. Okay? This shows you the third phase of this, the third aspect of aldosterone, of this process. Yes, with aldosterone, we're reabsorbing solute and water, but not just any solute. We're reabsorbing sodium, and at the same time, we are secreting potassium. So we're simultaneously regulating blood volume, blood osmolarity, and what's in the blood, the electrolytes. Because we reabsorb sodium, we'll secrete potassium to keep those two at the right levels. Okay? So aldosterone helps to regulate that. Plasma potassium does as well. If you have elevated plasma potassium, you're just going to have more potassium here. It's going to increase this process. And the amount of sodium in the collecting duct. If you have more sodium here, it will also increase that process. So they balance each other out. Now what aldosterone actually does is it increases the activity of these sodium potassium pumps. So if you have more of their activity, you will increase this process. All right. Now we also will secrete protons. There's two reasons that we will secrete protons. We're going to talk about the first reason here. This first reason, we're secreting the protons in order to reabsorb bicarbonate. There's a mechanism to reabsorb bicarbonate. It's rather convoluted. That's how we do it. Another reason we'll secrete protons is to directly get rid of them. Directly secrete protons into the urine. That's why the urine's acidic. We won't talk about that process just yet. We'll save that. That's what's going to be done in the intercalated cells. Okay? We'll save that until of the collecting. We'll save that until we talk about pH. Okay? So you notice this is happening in the proximal tube. And what we have here is a sodium proton antiporter. So sodium's coming in, protons are going out. We gotta have protons to do that with. And where do we produce them? We make them by breaking down carbonic acid into proton and bicarbonate. Where does that come from? Well, we know we can make carbonic acid by combining carbon dioxide and water. And as we know, that doesn't happen, that will happen normally, but it's very slow. We need that enzyme, carbonic anhydrase. So we have carbonic anhydrase here inside the cell of the proximal tube. So we're constantly making carbonic acid. Well, where do we need to make carbonic acid? We need CO2 and water. There's plenty of water around. CO2 comes from the blood, and it comes from the filtrate. So we take this carbon dioxide, we move it in here, we convert it to carbonic acid, we get a proton, the proton goes out. And what does that proton do? It combines with bicarbonate in the filtrate to form carbonic acid, which then makes CO2, because we also have carbonic anhydrase on that surface, on the apical surface there.
Mhm.
That should be associated with bicarbonate reabsorption. Okay. So the reason we are secreting this proton is so that we can combine it with a bicarbonate, then make it into CO2, move the CO2 into the cell, make it back into the bicarbonate, and the bicarbonate goes into the blood via this transporter here. Of course, once again, that all depends on the sodium gradient being maintained, and so we have the sodium pump to maintain that. Now, another way that this could happen is if we make the bicarbonate and the proton, actively transport the proton out, and then send the bicarbonate the other way. And that's what we're going to do in those intercalated cells, as we'll see. We also secrete ammonia and urea. This occurs pretty much throughout proximal convoluted tubule lipid down and collecting them. Now you'll notice we reabsorb urea and we secrete it. So what is this wishy-washy relationship we have with these nitrogenous wastes? Well, as we'll see, we're going to use them. Urea is more or less non-toxic. I mean, it builds up enough, but it's much less toxic than ammonia. So we can kind of let it hang around a bit. And as land-dwelling mammals, we have decided to use this process of letting the urea hang around a bit to help us reabsorb water. Because remember, as I indicated in the video, if you're going to reabsorb water, you've got to have some place of higher osmolarity for the water to flow into. Right? And so I've indicated that the medulla is maintained at high osmolarity. What is maintaining that osmolarity? A big part of it is urea. We're letting that urea hang around in the medulla to just be there to help to be an osmolyte, to help us absorb water. Okay? It's called ureoteliism. If you're a fish, you don't have this problem. Because you make ammonia by breaking down amino acids, and as soon as that blood with that ammonia gets to your gills, out goes that ammonia into the water. No problem. Okay? It's land-dwelling mammals without gills that have this, or land-dwelling animals without gills, and then animals that have conversely gone back into the aquatic environment after developing on land. All the other land-dwelling animals, the birds and the reptiles, they've just chosen to make an even less toxic form of nitrogenous waste, uric acid, which is a solid, right? And so instead of hanging out, keeping the urea around in their kidneys, they produce this uric acid that just forms in their cloaca and comes out as a solid mass. It's the white stuff in bird poop. If you have a pet snake, of course, it's the white mass in snake poop. Have I got a pet snake? Not terribly common. So how do we regulate this now? We've seen how hormones can regulate glomerular filtration, but what about the process of reabsorption and secretion? Well, these same hormones play a role in that too. So the renin-angiotensin-aldosterone system, antidiuretic hormone, and AMP all play a role in regulating the activity of these different parts of the tube as well. Right? So angiotensin 2, once again, it causes vasoconstriction of arterioles. That's not related to filtration or reabsorption and secretion directly. We know that that affects it indirectly by regulating filtration. But it also stimulates that sodium proton antiporter that I mentioned earlier. That increases sodium chloride and water reabsorption. Right, so the sodium comes in, chloride comes in with it for electrical balance, or comes across with it. And then of course the water follows the osmolytes. And of course its main thing is that it stimulates the release of aldosterone. As we've already discussed, aldosterone stimulates reabsorption of sodium and chloride in the collecting duct, as well as the secretion of potassium. And that increases the blood volume without changing its osmolarity, because of course, along with that sodium and chloride comes water. Right? This is nothing new. There's a couple of new facts here, but mostly this is stuff we've seen before. ADH, one of its principal functions is it regulates the water permeability of the cells that pass through the medulla in the distal collecting or distal convoluted tubule, and particularly the collecting duct. Here this is known as facultative water reabsorption, meaning that we can vary it. The water reabsorption in the proximal tube was the word I used? Obligatory. Obligatory water reabsorption. The reason is the aquaporins are always there. Here we can take the aquaporins in and out. And that's what ADH does. If ADH is present, antidiuretic hormone, diuresis means to increase the volume of the urine, right? Antidiuresis, we decrease the volume of the urine. Why? Because the water goes across those cells and back into the bloodstream, right? So by increasing the water permeability of those cells in the collecting duct, we will reabsorb more water. Now we'll see this a little bit later, and you'll be able to see how that works. Okay, and then AMP, of course, mainly works by interfering with these other ones. It reduces the effect of ADH, angiotensin 2, and aldosterone. We know, though, it has a direct effect on GFR. We discussed this earlier because it causes those mesangial cells to relax, increasing the filtration membrane area to raise the GFR. And of course, all that is going to decrease blood volume. All right, so now let's get into why the nephron is shaped the way it is, why the medulla has a concentration gradient or osmolarity gradient, why we have this pyramid-shaped arrangement, right? Everything's like a pie slice. At the cortex over the top, then the medulla, which steadily decreases in volume until we get to the peak. Right? Why is it arranged that way? It's so that we can vary the concentration and volume of our urine. As land-dwelling animals, one of our main concerns is drying out. How do we preserve water, but at the same time eliminate these soluble wastes that we have to have some water to carry out of our body? And as we've seen, regulate mineral contents and so forth. We've got to get rid of some water, but how do we hang on to as much as we can? And let's say that we're chugging down water left and right because we came to a stream or turned on the faucet nowadays. We think about it in modern context. And we have too much water. How do we get rid of that, right? So if you drink enough water every day and not diuretic substances like Diet Coke or Diet Pepsi, you're gonna have clear urine. Won't even look like urine, it'll look like water if you drink enough. Right? But then maybe you've been working out in the yard, or you've been drinking a lot of coffee and not drinking enough water, and then you go to the bathroom and it's not as much, and it's very yellow. Right? How do you do that? How do you change the concentration and volume? That's what we're talking about here. So let's first look at the instance where you're going to make dilute urine. You got plenty of water, you drink a lot of it, so you need to get rid of some water. So, you're going to form filtrate in the Bowman's capsule. It will then pass down the proximal tube. Now there, nothing will change. Nothing's different. You're going to reabsorb a ton of solute, all the glucose, amino acids, all that stuff, as I mentioned, it's going on the proximal tube with its huge surface area, and you're going to reabsorb a ton of water. Just because you have drunk a lot of water doesn't mean you're going to stop reabsorbing water. Because remember, you go from 180 to 178 maybe, instead of 179. Right? So it's maybe a liter change. Now we get from the cortex and we start to enter the medulla. The cortex osmolarity is isotonic, meaning it has the same osmolarity as any other body fluid, which, if you want to put a number on it, is about 300 milliosmoles. So all the water that we're reabsorbing there is due to the fact that we're moving all that solute as well. So we're moving water and solute, bringing a whole lot of volume across without changing the osmolarity. But now we start to enter the medulla. And we know the medulla has a osmotic gradient. The tissue of the medulla is near isotonic, right at the boundary with the cortex. And then as we go deeper and deeper and deeper into the medulla, it gets more and more concentrated. Notice this is labeled renal pelvis. That's the name for that, all those urine collecting structures. So we've reached now the peak of the medullary pyramid, and we see it's very concentrated. As we begin to descend via the loop of Henle into that ever more concentrated tissue, what we note is that the only thing moving is water. Remember, this is the portion of the loop of Henle that has simple squamous because it's passive transport. The only thing we have in there, more or less, is aquaporins. So there's tons of aquaporins there, and so as it flows down, the water moves by osmosis. Why? Because this is high osmolarity over here, and the water's coming out. So we're reabsorbing quite a bit of water there. And this filtrate is becoming more and more concentrated. And so when we reach the peak, ideally, that filtrate has reached the same level of concentration as that surrounding tissue. Now, we start to go up the tube. The nature of the wall changes. There's no more aquaporins on it. Initially, we have passive transport of sodium and chloride, simply because there's a lot here, there's less here, and so it starts to go out. But then as we reach higher levels, the gradients are no longer favorable for that, so we start to actively transport sodium and chloride. That's when we go from a thin tube to a thick tube, because we need that room. So now, the filtrate is becoming less and less concentrated. That process continues as we move through the distal convoluted tubule and the collecting duct. Ever more sodium and chloride is moving out. Notice we're not moving any water out at this point. And we end up at the end with a relatively large volume of dilute urine. The last place we reabsorb water was in the descending limb of the loop. From then on, it's all about transporting the solute. So we end up with a dilute urine with a relatively large volume. We put some numbers on it. Again, Your proximal tube has about 300 milliosmoles. I really need to get new versions of these. This is quite old. You guys were I'm like born with this, but it hasn't changed. So, okay, 300 milliosmoles. And then as it descends, it becomes more and more concentrated. And here we're looking at an example that would occur in a cortical nephron. In the next example, they're going to show you juxtamedullary nephron, but it's the same deal. Remember, the papillary duct, and that's what I don't like about this drawing, the papillary duct or collecting duct is going to go all the way out, okay? But the loop of a cortical nephron is only going to go down part way, okay? So it's going to get concentrated as the water moves out, and it reaches the same osmolarity as the surrounding fluid, right? That's what's happening here. Then it's going to ascend through. We're not long moving water anymore. We're moving solute. And so it's getting ever more dilute, ever more dilute, ever more dilute, and we end up with dilute urine. All right? So that's if we got plenty of water, we need to get rid of water. Hang on to solute. So to summarize the formation of dilute urine, first we reduce the volume, or let's skip the proximal tube, right? Because that's going to be the same no matter what. Most of the reabsorption is going on there, most of the solute, most of the water, okay? What matters is from then on, right? So we go into that loop of Henle, we reabsorb water, the filtrate becomes more concentrated, and then we initially have diffusion, and then active transport of these sodium chloride and other solutes, mainly sodium chloride though, as we now move throughout the rest of the tube. In the absence of ADH or minimal amount of ADH, the collecting duct is relatively impermeable to water. There aren't many aquaporins there, and so the final outcome is we have a large volume of urine with a lower concentration than our body's fluids, right? If you drink too much water, you're diluting your whole body, so you need to get rid of that water. And so what you secrete will be a lower concentration in your body. Okay? Now, what happens now when you haven't been drinking enough water? What happens? Well, initially nothing changes. Again, what's happening in the proximal tube is the same. You're just reabsorbing most of the stuff. What's happening in the descending limb is the same because it's passive. You're reabsorbing a lot of water by pushing that filtrate into that high osmolarity medulla and allowing it to reach equilibrium, osmotic equilibrium. Ascending limb, not much is the same, or nothing's really changed. Initially you'll have diffusion and then active transport. Right, and that continues. It seems to be making dilute urine in it, but now we reach the difference. We're going to go back through the medulla. And when we go back through the medulla, we're going to put aquaporins in there. So it's going to be a similar process to what's happening here. As we move a lot of water, as we pass through the medulla again, the urine becomes ever more concentrated. And we end up with a low volume with a higher concentration than our body's fluids. How high can it go? As high as the peak of the renal pyramids, or the medullary pyramids. Okay? So the only difference here then is the permeability of the water in the distal convoluted tubule and the collecting duct. Here you can see the numbers on it again. Notice they're showing you a juxtamedullary nephron this time. So initially it's 300, and as we pass into the ascending limb, we're moving just water. If we go all the way down to the peak, it reaches a peak concentration of about 1,200 milliosmoles. As humans, the length of our pyramids, right? If you look at animals, how much can they concentrate their urine? What matters is the relative thickness of the cortex compared to the length of the medullary pyramid. And with a human, you can concentrate to about 4 times. If you're a kangaroo rat that lives in the desert, you can go much higher than that. Okay? That's why you don't ever really have to drink. So, goes down, becomes very concentrated. Then you start to pump out, or move, and then initially pump out the fluid, or the solute, so it becomes more dilute. And after that, we start to now move water again as it passes down through the medulla once again, and can reach a concentration of about 1200 milliosmoles. Now this image also tells us part of the story of how we have the osmotic gradient. The urea being reabsorbed here and secreted here tends to stay in that medulla. So we use that urea as an osmolyte to help us concentrate our fluid. The vasorecta, that ladder-shaped vascular formation, helps keep both salt and urea in the fluid, because as the water is reabsorbed, that creates a concentration gradient that stimulates the salt and urea to flow from one side to the other. Okay? And that helps to keep those osmolytes, urea and the salt, in the medulla to maintain that osmotic gradient. So to summarize the formation of concentrated urine, again, nothing changes initially. Proximal tube is the same. Descending limb is the same. Reduce the water volume, water reabsorption, filtrate becomes more concentrated. Ascending limb is more or less the same. We have initially diffusion and active transport of sodium chloride. Now the difference is the ADH is present. The collecting duct is permeable to water. So as the filtrate passes once again through the medulla, we're reabsorbing a lot of water. As we mentioned, there's also that recycling of urea and salt. The final outcome, you have a small volume of urine with a high concentration in the beginning, scant urine, as we said. And it's high, how high? It can be up to 1200 milliosmoles, 4 times more concentrated in your body's normal osmolar. This is just showing you the main difference is going to be that transform. Okay. Now, what I want to point out in this slide, though, and this makes it really apparent because it's not cluttered with other stuff. If you look here, you've got 2 different processes going on the 2 sides of the loop of Henle. On one side, you're transporting water. The other side, you're transporting salt. And those 2 sides are connected together. This creates what we call a countercurrent multiplier, which is the other part of the explanation as to how we produce and maintain the osmotic gradient in the medulla. Countercurrent mechanisms are extremely common and important in biology. One example that you might have heard of is the countercurrent heat exchange mechanisms that many animals use to keep their limbs from freezing, but at the same time keeping their body's heat from being sapped away by their limbs. When I think about a penguin standing on ice, its feet are, they're a part of dinosaur feet, right? They are scaly, they don't have fat on them. So what keeps the feet from freezing? And what keeps the heat from being sapped out of the body's core by the heat, by the feet in contact with the ice? Well, the blood vessels of the penguin's circulation are arranged as a countercurrent heat exchanger. The arterial blood flowing into the feet runs very close to the venous blood returning from the heat. That venous blood is going to be cold. And so what will happen then is the heat will go from the artery into the vein before it even gets to the feet. And so you'll have already cooled blood that's circulating through the feet, and so it doesn't carry the heat out of the body's core. Carries just enough though to keep the feet from freezing, but not so much that the penguin can't maintain its core body temperature. Of course, it has a big blubber layer to help it with that. And then the chick stands on the feet, and the gut of the penguin folds over the chick to keep it warm. It's very cute. All right, so this is what we have, a countercurrent mechanism, countercurrent exchange. You do the same thing with heat exchangers in houses. It's not such a big deal here in the South, where we don't have a huge problem with cold. I mean, it gets cold, it's not like Chicago cold, right? But if you have a house in Minnesota built to modern standards, they're gonna put, huge thickness of insulation on it. It's gonna be sealed up tight as a drum. And so how do you get fresh air in there in the wintertime? Well, you have a ventilator that's bringing that Arctic freezing air in from the outside and pumping warm that you paid to heat, stale air out. Well, how do you keep from freezing to death with that? Well, those two air streams go past each other in a heat exchanger so that warm heat that you've paid so much for, a good portion of it's going to go into the cool air that's coming in from the outside. Now the multiplier comes from the fact that these two are connected now in the loop of Henle, and we have active mechanism on one side. This is what's depicted here as a passive mechanism. Okay? How do we put this into an active mechanism? Well, to help us understand that, we need to look at a video that I hope is still there. If not, I will find another one. Let's see. Here it is. No, page not there. Okay. So let's see if we can do it. Yeah, I think this is it. Download it as a move. Not maybe 34 minutes for it. Yes, it is. Okay. Now, the magic happens when we turn on the fluid flow. We're going to push that 400 around, and that 200 that we made up. So it's 200, 200, 400, 400. See what we're starting to do? Now we repeat. Turn on the pump. That's 300 there, 200 there. We're going to pump it to about 150, 250, 300 to 500. Turn on the water, let it equilibrate, pump it around. And as we keep doing that, we're going to progressively produce that gradient going down. Okay? This constantly happens, and it relies on the difference in the permeability here. This is only permeable to water, this is only permeable to salt, and the fact that we are actively transporting salt. Right? That's where we get the multiplier. That active transport multiplies the effect of that countercurrent exchange to produce a gradient. And again, the gradient is dependent on how long that loop of Henle is. All right. Now the last topic is, what do we do with that completed urine? And I think this is something that we can cover better in lab, because I will have talked about the anatomy of the ureters and the bladder then. So I think that is what we'll stop with today. Now, we'll have a video over the weekend, it'll probably be about 45 minutes long, covering this last topic, which is going to be fluid balance and pH, okay? This is a summary topic, really. Now, it's going into a little bit more detail about water input and output and pH and electrolyte balance, but it's mainly a review topic, because it's going to be hitting these same things about ADH and aldosterone and AMP and all this stuff we've seen a bunch of times, right? So you can watch that on your own, and so that means Monday, all we'll have left is review. So we'll do some review, we'll have a quiz on it, we'll do some review activities, and then any other questions you have will be freely available for that. Okay? So today in lab, we'll do our last part, which is on the urinary system. We'll look at some of the anatomical details here. Okay? Now, it doesn't take me very long to cover it. So if you want to go in and start at 1:30, you can, but I'm not going to come in there and begin my discussion until about 15 till, because we're having our potluck lunch today for the department, and I want to wolf down a few of the delicious things and then come in at about 12:45 to start our lab. Okay, so we'll start lab in about an hour. But if you want to go in there, of course, and start working, you can, reviewing stuff from last week or on the lymphatic system, which is not much. We're going ahead and getting a head start on the kidneys, but I'll go in and do my introduction and answer any of your questions beginning at let's see, 1:45 or so. Okay. And the urethra. When we look at it in context, what we note, of course, is that the kidneys are kind of up in the superior part of the abdominal cavity, and that the right kidney is lower than the left. Of course, this is due to the space occupied by the liver on the right side. If we take a view of the transverse section, we get a little bit more context. We note that the kidneys are and the entire urinary system actually is retroperitoneal, right? It sits behind the parietal layer of the peritoneum up against the posterior abdominal wall. You may recall in the cats, when you grab all the intestines and pull them aside, the kidney's still there sitting on the back. And if we had a fresh cat, if we looked at a fresh cat, you can actually still see that as you begin to dissect it, that parietal peritoneum, shiny, almost like plastic wrap up against the posterior abdominal wall. And then there's this bump there, and that's the kidney. And the reason it doesn't look like a kidney, looks like a bump, is because it's covered with fat. There's an adipose capsule surrounding each kidney, and then a renal fascia around that. And that entire structure is in contact with the peritoneum, as you can see here. There's also the adipose, or the renal capsule itself, which is the outer connective tissue surface of the kidney. Now, in this, we see this is the body of L2, and we can see now the fact that we can see a bit more of the left kidney, because we're looking sort of at the top of the right kidney here. Go back to this, kind of right through there, and then we're sort of the middle of the left kidney, and you can see the fact that, as I mentioned in the lecture, that you've got this light-colored cortical material, and then the dark-colored medulla, divided up into those pyramid structures. The pyramids have their bases facing the cortex, and then they taper towards the medulla, where we see this space. Okay, now that space is called the renal sinus. As we'll see, it's not empty, it's got a lot of stuff in it, but that's what we're seeing here, is that renal sinus space. We can also see how the kidneys are protected by the bodies of the vertebrae and also by the vertebral ribs. If we look here, you can see the portions of the ribs there. They're protected. Okay, here we can see a view of the kidney with all the relevant structures Again, the outer part is a connective tissue capsule. And deep to that we have the renal cortex. Okay. And then the renal pyramids that make up the medulla, with their base up against the cortex and their peaks pushing into the renal sinus. And as their peaks push into the renal sinus, we call them papillae. There's a renal papilla. And the completed urine, of course, drains from the papillary duct or the collecting ducts that exit via that papilla. Now, one thing I didn't mention in the lecture, the video lecture, which is fairly obvious here, is that we have these columns of cortex-like tissue going from the renal sinus up into the cortex, these renal columns. And these are allowed to allow a region of normal osmolarity for the blood vessels that are coming in from the renal hilus, right? We see it there, or hilum. I always forget which ones call it hilus, which call it hilum. The indentation of this bean-shaped organ, and with other bean-shaped organs that have that central indentation, we call a hilus or a helium or a hilum. That's where all the blood vessels go in and out, right? So the artery and vein is coming in there in the middle, and they have to rise up into the cortex, because we know that's where the glomeruli are going to be, and the renal corpuscles. And so we have to have some way to get them through the medulla. And so that's what these renal columns are about. They are carrying those blood vessels up into the cortex. Now we also note that the renal sinus is not empty, it's full of stuff, right? What's most notable is this large structure called the renal pelvis, which is the urine collecting structures. Each of the papillae has a structure around it called a minor calyx that is almost like a funnel that collects the urine that exits from that papilla. You can see it there. There's one, there's one. Several of these minor calyces will come together to make what is known as a major calyx, sort of the common collecting point for several of them. And then they finally drain into the larger portion of the renal pelvis itself, which we note exits the kidney at the hilum and then tapers and becomes the ureter. So we have all these urine collecting structures that taper and become the ureter. Now, as we note, there's also blood vessels going into that renal sinus, the branches of the artery and vein, and then any remaining space is filled with adipose tissue. Right? Again, that's one of the functions of fat, is it fills, it's like the packing peanuts, it fills in spaces. And here they're showing you the orientation of a nephron with its, this is a juxtamedullary or cortical nephron with its renal corpuscle up, and then proximal tube, loop of Henle, distal tube, and then collecting duct, right? And again, each of these is basically a bunch of these nephrons, and I use the analogy of a bunch of flowers. If you hold the base of a bunch of flowers and they spread up and out, that's sort of what we have here. So if the renal corpuscles are the buds of the flowers, they're up here. Okay. What it also means is we have more or less a functional unit, a lobe of the kidney, which would be from the center of one column to the center of the adjacent one, encompassing one pyramid and the cortex that overlies it. The reason we would consider that one unit or one lobe is because all of the nephrons are going to drain through the same papilla. Right? So all the nephrons that are arranged here are going to drain into this papilla, and all the ones that are over here are going to drain into that one, and so forth. Okay. Now I won't do this because we've already gone through this in quite a bit of detail. Sometimes I have this lecture before the lecture and lab starts, so we won't go through them. This is a nicer picture of the vasa recta than the one in the slides that we have so far. You can see how the juxtamedullary nephrons loop of Henle goes all the way down. The cortical doesn't. The dotted line represents the boundary between the cortex and medulla. Okay, and we've seen this picture of the renal corpuscle. You guys have noted them here. We've also looked at the structures that make up the filtration membrane, the fenestrated epithelium, the basal lamina, and then the visceral epithelium. And there's a nice electron micrograph of it. You can see the capillary endothelium here with the fenestrae and all the interconnected fibers. They call it the basement membrane here, but it's a basal lamina. The difference is that a true basement membrane also has a reticular lamina from connective tissue. Here, all we have is two epithelia. This one, the podocyte epithelium, and this one, the capillary endothelium. And those together make their combined basal lamina. Yeah.
The processes, aren't those just the pedicels?
Yeah, these are the pedicels.
Okay.
Foot process is what pedicels. And then we have the filtration slits in between them. Okay. All right, and I also went through this, showing you the difference in the histology at different points along the nephron. Okay. So one thing we need to talk about is the blood supply to the kidneys. Now, we haven't talked in detail about blood supply to individual organs except perhaps the heart. We went into the brain, the arterial circle of the brain a little bit. The kidneys actually receive a great deal of blood for their size. So it's important that the blood supply be especially well organized here so that we can push that large amount of blood through this relatively small structure with minimal resistance and get it a high blood flow through the kidneys. So of course this begins with the renal artery and vein, and we ultimately have to get that distribution up to each renal corpuscle, form a glomerulus, and then of course we have to have the blood, we have to have the peritubular capillaries to receive the reabsorbed material. Okay, so here we can see a kind of a diagram of how this works. Now this is a picture of a triple injected pig kidney. It's been sectioned sagittally, and then a little bit I'll pull some out of the buckets and we can look at them. It's very simple dissection. But what we see, of course, it goes from the aorta to the renal artery. Okay? And then usually there's a few segmental arteries. This is somewhat variable. And then that goes into the lobar arteries. And we'll look at. Okay. Now, where it really gets interesting though, or gets consistent, is the interlobar arteries. The interlobar arteries are the ones that rise up through the renal columns. Okay? And then what happens is the interlobar arteries form an arterial arch over the base of each pyramid. So I can see that. Yeah, you can see that here. So imagine down here is the medulla, up here is the cortex, and this is an arcuate artery running right along the junction between the cortex and the medulla. You can see those up there, kind of turning in. And then from those arcuate arteries, we have interlobular arteries. An interlobular artery rises up from the arcuate artery and will serve a number of nephrons. So a lobule would simply be the nephrons that each one of these serves. Okay? Interlobular artery, interlobular vein. Don't get that confused with interlobar. The interlobar ones go up the column. Then each interlobular artery gives off the afferent arteriole, which then goes and forms the glomerulus, of course, and then the efferent arteriole, and then the peritubular capillaries and vasorectum, if we're talking about a juxtamedullary nephron. Right? And then that drains, and it's variable. It can drain through interlobular veins, as we see here. It can drain back in there. And in some cases, it goes directly into the arcuate vein. And then interlobar back to the columns, and then finally down the renal vein. Okay, so you can see we've distributed that blood in an organized fashion to each of the million nephrons or so that are in each kidney. All right, we also talked about the the tuboglomerular feedback. I don't know if I use this term, but that is sometimes called the juxtaglomerular apparatus, the structures involved in that, okay? And that consists of the juxtaglomerular cells we see around each afferent arteriole, and the macula densa, which is around the distal tube or the loop of Henle where it comes back by the renal corpuscle. And again, it's the communication between these two that is responsible for the tuboglomerular feedback, as we discussed today. Okay. Now, so we understand now the structure of the kidney and the structure of the nephron. Once that completed urine is collected in the renal pelvis, of course, then it will drain into the ureter. And the ureter, as I think I said before in class, is not simply a drain tube. It is has an active wall. Okay? It has a mucosa and a submucosa, and it has a muscularis, just like the walls of many other hollow organs, including your digestive tract. Okay, there's a difference though here. In this case, the circular fibers are deep. The longitudinal fibers are deep, and the circular fibers are on the outside. They're superficial. So it's just a difference in order. But the point is that we still have longitudinal fibers and circular fibers. And if we have longitudinal fibers and circular fibers around a hollow organ, what sort of motion can we do?
Peristaltic, peristaltic.
Peristalsis, exactly. So we have peristalsis waves going down the ureters, and they are essentially pulling the urine out of the renal pelvis and actively pumping it into the bladder. Why it can happen when you're hanging upside down. Hopefully you're not doing that very long. Okay, when you're laying down and when your bladder is nearing being full, you can shove some more urine into it. Hopefully nobody urgently needs to go right now. I used to torture my sisters. We were on long rides and they'd say, "We have to pee. Oh, it's a few more miles." And I'd say, "Oh, think of flowing streams and waterfalls." That was a very nice brother. Anyway. Now, one of the other features we see about the ureter, and this is also true of the renal pelvis and true of the bladder, is that it's aligned with the transitional epithelium, right? And that's something we talked about in AP1, that we have this epithelium that is multi-layered. It's a stratified epithelium, but we don't assign a shape to it because its shape changes. As it comes under pressure with the urine, it's going to flatten out, stretch out, and flatten out. And then when it's relieved of that pressure, it will rebound, and the cells become more, they're close to cuboidal, but we don't really call them that. Okay, so that's going to line the renal pelvis, it's going to line the ureters, it's going to line the bladder, and the very initial segment of the urethra just before the sphincter. Anything that's going to come under persistent pressure of stored urine. Speaking of the bladder and the urethra, you can see it here, the urinary bladder. You should say urinary bladder, just distinct from the gallbladder. Its wall is also made of smooth muscle, but it's not really going to do peristalsis. Instead, it has to just basically squeeze down and eject the urine when you urinate. And so what we have is this lining, of course, with rugae on the inside of it. Those are, of course, going to be stretched out when the bladder is full. And this muscular wall in the bladder, this is known as the detrusor muscle. That's the smooth muscle that makes up most of the wall of the bladder. When that contracts, it will eject urine from the bladder. Now, when it's relaxed, it can easily expand. Remember, muscles can stretch without damage. That's one of the features of muscle. And so as the bladder fills, this will expand like a balloon when that muscle is relaxed. Now, that will kind of crush the opening of the, not damage, but kind of collapse the opening of the ureter to kind of act as a valve to keep the urine, once it's kind of shoved into a mostly full bladder, keeps that pressure urine under pressure from backing back up into the ureters. And if we see the opening of the ureters are here on the floor of the bladder, as is the opening of the urethra. And this is a smooth part. This part doesn't expand. Okay? It's like if you took a balloon that wasn't blown up and you glued the bottom of it, or just laid it down flat on the table with the kind of the tube off the side of the table and you glued it down. Well, the part that's in contact with the table that's glued down is not going to expand, but the part above it will. So if you were to blow it up, it'll kind of blow up like a dome, and that's what happens here. This trigone or trigony, I've never seen a good analogy of that. This is this smooth portion of the floor of the bladder where between the ureters and the opening of the urethra, and that part does not expand. When people have persistent bladder infections, this is the part that's often harboring the bacteria. All right, okay. Now, the urethra opens from that and into, of course, then drains out of the body. And, of course, we have sphincters that control the exiting of that. One portion of the wall of the bladder, right around the opening of the urethra, acts as a smooth muscle sphincter. So this is known as the internal urethral sphincter, and it acts to control the movement of the urine in an involuntary fashion. That's smooth muscle. The urethra then passes out of the urogenital diaphragm, the pelvic outlet muscles, and there those muscles will wrap around the urethra and act as an external urethral sphincter that we control voluntarily. Okay? Now obviously this is much longer in the male than the female, and the lining of that urethra changes a bit. It's mostly just a simple columnar epithelium, though it does vary a bit in the male in becoming squamous, stratified squamous at the very end. And we of course know, we've talked about how the urethra goes through the prostate, so that if you get hyperplasia of the prostate, it can interfere with urination, because note this is before the diaphragm. So even if you open up that diaphragm and your prostate is swollen and constricting your urethra, you're going to have a hard time emptying your bladder. And a lot of men have trouble with that, especially as they get older. It's quite very common. It doesn't even necessarily mean you have prostate cancer, just benign prostatic hyperplasia. All right. So here we can see, of course, the difference in the length there. The last thing I want to talk about was the last slide we had today, but I thought it would make more sense to talk about it here because I had just mentioned those smooth muscle sphincters and the skeletal muscle sphincter. This is the micturition reflex. Micturition is the fancy word for the process of urinating. And it's controlled in the pons via both the autonomic and somatic motor systems. You got two centers in the pons that help with this. The pontine storage center that promotes the activity that's going to result in storing urine, and the pontine micturition center, which is going to result in the activity that results in micturating or urinating. Okay? So let's say that your bladder is filling, you're not ready to urinate yet. What that means is you're going to have a very low stretch read by the visceral fibers coming from the pelvic splanchnic nerve from the bladder. Okay, that's detecting the stretch of the wall of the bladder. That is going to stimulate the lower motor neurons that innervate the fibers that form the external sphincter. So this is one of your skeletal muscles that's going to contract fairly consistently, even in the absence of you actively contracting it. You don't have to concentrate to keep your bladder from emptying unless it gets extremely full, right? Then you're kind of squeezing your knees together like a little kid doing the pee-pee dance, right? But other than that, you don't really have to concentrate on it. It's a reflexive contraction of that muscle. This also stimulates the sympathetic preganglionic neurons that are going to innervate the bladder ultimately. And when the bladder gets sympathetic stimulation, that inhibits contraction of the detrusor muscle, and it stimulates contraction of the internal sphincter. So you relax the detrusor muscle and you stimulate the two sphincters, that equals storing of urine. So the ureters can now pump urine into the bladder and it will not yet empty. Yes?
What about people that get scared and pee themselves?
I think what happens in that case is the micturition center is activated by the emotional response to that, right? And it makes sense because if you're about to run, you don't want to be weighed down by extra urine or something, so you just kind of dump it and go. Okay, so now as the bladder fills, the situation changes. The detectors start to detect high levels of stretch. That will then stimulate the pontine micturition center, which in turn stimulates the pelvic splanchnic nerve here, which is the parasympathetic output, you recall, to the bladder. The parasympathetic input of that acetylcholine there will stimulate the detrusor muscle and inhibit the internal sphincter. At the same time, it will now inhibit the lower motor neurons of the external sphincter. So now we relax both sphincters and we constrict the muscle. Notice they also inhibit the sympathetic output. Once again, whenever you change autonomic innervation to something, it's never all in one. You go down on one and up on the other. Okay? Now, a little baby, this is all they can do. This is reflexive, and it doesn't work any other way. Okay? As you get older, you develop the motor pathways that allow you to consciously choose when this is going to happen, right? It's this input from the storage center that develops. So if that's not present, then this is more or less relaxed, and you rely on the input, the changes in the internal sphincters condition that changes. So the bladder fills, empties, it fills, it empties. There's nothing that can control it. Fairly early on in development, that pathway develops. And so now the baby, the young infant has the ability, theoretically, to control when they urinate. But they don't yet until they get quite a bit older, because it's a cognitive process, right? Up until that point, I feel that urgent bleed, I'll just go, right? They don't understand that there's an appropriate place for that. And if you learn to do that, you don't have to wear diapers anymore, and that's a desirable thing, right? It's the normal process of maturation. And of course, that can take a while before you stop going to your bed at night, nocturnal emesis and things like that. But that is the basic process. It's a sympathetic or an autonomic reflex that is partially overridden by automatic somatic motor output from the pontine storage center. All right, so let me show you the slides and models. Okay, so here's the urinary system. All right, now you guys have seen kidney slides before. These are really nice ones though. This is bigger. I'm guessing this is a guinea pig or something. If you'll look at closely at this, you'll see it has one big renal pyramid, which would be typical of a small mammal like that. So if we look here, we're in the cortex. There's your capsule. Here we can see a number of renal corpuscles kind of in a row. That's going to be a collecting duct in the center there. And there's another one. There's another one. There's an interlobular artery right there. We can just see a little bit of it. But we've seen all this stuff before. Let's see if I can find one that shows you the juxtaglomerular apparatus. Ooh, there it is. Right to it. Right, so here we can see a really nice glomerulus, and there's the Bowman's capsule, and there's the distal tube, and look at how closely packed those cells are. That's the macula densa. Went right to one. Find another one. It's not quite as good. And there's a good one. Yeah, you can really see the macula densa right there. See how densely packed they are? All right, now if we go down into the medulla here, we of course see a bunch of collecting ducts and loops of Henle. Can't really tell the difference. If we were to look carefully, we might find some that had simple squamous epithelium. Those would be descending limbs, like some right there. They've been collapsed though. And down here, they kind of look like circles. That's because that's where they're turning around. Here we can see some thin loops of Henle down there. You can see some simple squamous down there. And then this will be your renal pelvis space down here. You can see it's got that transitional epithelium. That's the kidney. Those are really nice slides. All right. Here's a ureter. Not much to see on these. They're kind of faint slides. This is like a little rat's or mouse's ureter. Oh, this one's kind of light, but you can see that. That transitional epithelium there. There's your submucosa. You can see the different muscular layers. All right, so that's a ureter. It's very nice. And then we have bladder that's smooth. That's relaxed bladder, nice transitional epithelium. And that they stretched it before they put it on the slide, so you can see how the shape of that epithelium changes. All right. So those are the slides we have. Now, if we take a look, I see some of you have already found the models. We have a number of copies of this model here. Very nice German model. But it shows you
Yeah, I didn't know they.
3 levels of detail.
I think they were.
You've got the entire kidney, which is a good place to find the urine collecting structures there. This shows you a single pyramid, or a close-up of a single pyramid. There's your interlobular artery coming up, your interlobar, your arcuate artery and vein, your interlobular, and a set of corpuscles. They've got detail of one with the proximal tube, loop of Henle, distal tube. They've got the cortical and a juxtamedullary here. On this side, they show you the vasa recta as well. And then this one shows you a very nice detailed model of renal corpuscle with the proximal tube, the distal tube with the macula densa, the afferent arteriole with the juxtaglomerular cells. The glomerulus, they show you podocytes on one half of it, but not on the other. Okay. Capsular space, this is very, very, very nice. Okay. We've got a number of other larger models of the kidney as well. And then we've got this one. This is my favorite model. We call it the Great Tome of the Kidney, the Great Book of the Kidney. You open it up and we read about the kidney and then we close it. It's more or less useless. It's so undetailed, so useless. I just think it's funny, so I keep it around. This is the Great Tome of the Kidney. So that's the main models we have. And then of course you can find kidneys on any of these Corso models. Probably larger than the human kidney, which would be more like that. Okay. And of course we can see, even without the latex, the difference in color between the cortex and the medullary pyramids, which are depicted as being quite a bit smaller on this. Okay. You can also see the renal columns in between them. And what they've done is dye the arteries and veins blue and or red and blue. And they also tried to dye the urine-containing structures with the yellow latex. And you can see a bit of it here, but it didn't solidify. It's just all crumbly, so I tried to wash most of it out. Okay. Now this is actually a cyst in the renal. This is a renal cyst in there, which are fairly common, not necessarily a problem. There's some more of that cured latex. Okay, well we can see any of these renal pyramids, you'll notice there's a little cup around them. That's the minor calyx. And if I kind of push through there, it goes into this bigger space. That's the major calyx. All right, and then ultimately it all goes into here. There's the ureter. If I push through from there, I'm hitting a blockage of some kind. That's the artery. That's not wanting me to feed through there, but you can see how those urine-containing structures ultimately lead outside of the body or outside of the kidney. I might be going into the renal sinus there. That's not letting me poke through. Anyway, that's just the basics of it. See the minor calyces, you know, if you put three together, you got the major calyx and the renal pelvis. And of course, if we were to pull all that out, I can kind of do it here if I kind of reach in there, and you can see it's just a space inside the kidney. If I was to pull all that out, there's a space inside the kidney, an empty cavity. That's the renal sinus, and that's where all those structures are, including the adipose tissue. And then up here you can see some of the interlobular vessels going up, right? They're pretty small, but you can actually see that the latex filled them. And the interlobers go in between in the renal columns. It's pretty straightforward. Looks just like the picture. It's one of those few examples where it does.
We got the renal capsule. The hilum is the entry point.
Now cortex and renal columns.
From what I know, it wasn't these right here, the renal columns. Oh, they're the ones that okay, gotcha.
Now we're looking at the medulla, renal pyramids and the papillae.
Okay, renal pyramids right here, and then the papillae right at the bottom.
Renal sinus.
You can't see it on this
But
thing, but it would be kind of ear on the side somewhere right there. Yeah.
Renal pelvis.
Renal pelvis is this big area right here.
Major calyces.
Major calyces right here, and then it goes into the minors. Yeah, we'll skip that.
Alrighty, there.
We can't really.
Can't show the veins. Yeah, we
Not
can't do that.
really.
We have to do that by ourselves.
Renal.
Renal corpuscle. What makes up the renal corpuscle?
That whole thing right here, pretty much.
It's going to be the
It's the
afferent
afferent. You get the afferent, the efferent, and the. What's that?
glomerulus.
The glomerulus. Glomerulus.
Close enough.
Capillaries.
And then the Bowman's capsule.
Bowman's capsule. This whole area around it.
Mm-hmm. Then podocytes.
Podocytes. These right there,
Mm-hmm. And filtration slits.
which are in between them.
Yeah. And then.
We already got the capillary space, which is the capsule space right here.
Mm-hmm. Filtration membrane.
Membranes in there too.
Okay. Proximal convoluted tubule.
Proximal. Where are we at here? So we have the proximal right there. Leads out. We
Into
go
the
down
loop of Henle.
the loop of Henle.
Descending and ascending.
Yeah, descending and then ascending. Then we go into the distal, into the. Well, I'm following the
collecting
line.
duct.
Yeah, this is collecting and then the papillary duct. But then you also have the capillaries here, which we have the vestibular capillaries and the. Is the vas?
This is the vasa.
It's a vasa recta.
is yeah, that's the one that goes
And then-
into the justa medullary.
And that's the cortical.
Okay. And then we. Where is this? Oh, these are these.
Macula densa.
Macula densa. These right here.
Justa glomerate.
Which
Yeah,
are
cells are
there.
yeah, the orange ones.
they're the ones that contract.
The mesangial cells.
Mesangial cells would be all up in here.
Yeah, and then there's also the adipose tissue.
Adipose tissue. We already covered those. The collecting ducts. And
Ureters, tertius epithelium.
we have the
can't see that.
ureter right there. Comes out. And that other, I don't think we can show that. I don't think we have a bladder. Right?
No.
No circulation. Start from the aorta, renal artery. Then you go to the
Segmented.
segmental.
My bad, please. I cannot segmental. My bad. Then the. Apparently from here it says the interlobar.
Lobar goes up,
Into,
which is also this one.
yeah, same thing. And then the flat ones right here are the arcuate.
Arcuate. And then the one going up is the interlob.
Yeah.
Interlobular.
It's kind of the same wave to the veins too.
Yeah.
So
And the interlobular connects to the nephrons.
Then it goes from the arcuate interlobular, and it goes afferent to the efferent. Then it goes downward to the interlobular veins, arcuate veins, interlobar veins, and then it goes to the vein.